This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas.
Imagine you have a population of engineered microscopic immune cells actively hunting down cancer inside your body.
Right.
And they are incredibly powerful.
Right.
Like maybe a little too powerful. You start feeling a severe fever coming on, which is a sign that they're attacking healthy tissue in the crossfire.
Yeah, really dangerous situation.
Totally. But to control them, like to hit the emergency brake and put those cells to sleep, you don't have to rush to a hospital for an expensive IV drip of immunosuppressants.
No, not
You don't need a complex surgery. You literally just walk into your kitchen, brew a normal cup of coffee, and take a sip.
It sounds like pure cyberpunk fiction, you know?
Yeah.
The idea that our biology could act as like hardware waiting for very specific, totally mundane liquid software update.
Right.
But we're looking at a reality where this isn't speculative anymore. It's actively happening in the laboratory.
And that is the mission of our deep dive today.
Oh.
We are exploring some truly groundbreaking chemical engineering that details the creation of what's called the Caffeine-Operated Dissociation System.
Which goes by the incredibly unassuming acronym COES.
COES, yeah. Unassuming is definitely the word for it.
I mean, when you think about the profound medical revolution it represents, COES feels a bit understated, frankly.
Okay, so let's unpack this for everyone listening. We are going to look at how scientists used cutting-edge artificial intelligence to engineer a microscopic, caffeine-triggered remote control for human cells.
Exactly.
So whether you are a synthetic biology nerd, someone following the future of oncology, or just someone holding a mug of coffee right now, this deep dive is going to fundamentally change how you view your morning brew.
Absolutely. And to really grasp the magnitude of what's been achieved here, you basically have to throw away the textbook drawing of a human cell.
Oh yeah, the one from high school biology.
Right, where a cell is basically just a water balloon with a few neat little organelles floating around in a clear liquid. The reality is wildly different.
It's way more chaotic than that.
Oh, it's an incredibly dense, chaotic metropolis. You have billions of proteins, enzymes, and molecules violently vibrating and crashing into each other millions of times every single second.
Like a microscopic, highly regulated mosh pit.
That is a very accurate way to visualize it. And in that mosh pit, biological processes like a cell deciding to divide or an immune cell realizing it needs to attack a virus, they don't just happen by magic.
Right, there has to be a physical trigger.
Exactly. They are usually triggered when two very specific proteins physically collide and lock together perfectly. They bind.
Okay.
In the world of synthetic biology, researchers have spent decades building a toolkit around this mechanical reality. It's known as chemically induced proximity, or CIP.
So in my mind, the analogy for CIP is kind of like molecular Velcro.
Okay, I like that.
You have two proteins floating around in the cell that normally ignore each other. They just bounce off one another. But then you introduce a specific chemical, a drug, usually, and that chemical acts like the sticky interface of the Velcro. It wedges between them, forces the two proteins to bind, and boom, the biological process kicks off.
And the field has actually become remarkably adept at engineering that kind of molecular glue over the years.
Right.
But the architecture we are dissecting today relies on the exact opposite mechanism.
So not inducing proximity.
Right. We are not talking about inducing proximity. The engineering here is built on chemically disrupted proximity, or CDP.
Okay, so sticking with the Velcro analogy, if induced proximity is using a chemical to stick the Velcro together, disrupted proximity is using a chemical to instantly dissolve a bond that's already there.
Perfectly said.
The Velcro is already firmly stuck together, holding some cellular machinery in a specific state, and the drug acts as a wedge that just tears it apart.
That physical tearing apart acts as an off switch, or sometimes an on switch, depending on what those two proteins were doing while they were joined.
Right.
But from an engineering standpoint, breaking a pre-existing bond is vastly more difficult than gluing two things together.
Wait, why is that? Because, I mean, intuitively it feels like breaking things is always easier than building them, you know?
You'd think so. But it comes down to surface area and molecular physics. When two proteins lock together, they share a very large, incredibly complex, interlocking surface area.
Like puzzle pieces.
Exactly. Think of two pieces of a jigsaw puzzle perfectly snapped together. Trying to find a tiny chemical molecule that can somehow wedge itself into that massive, tight interface and pop them apart it's chemically grueling.
Wow.
It's akin to trying to pry apart two industrial supermagnets using nothing but a wooden toothpick.
Oh man, that paints a really vivid picture. The connection is too tight and the tools are too weak. So we need better toothpicks.
Precisely.
Which is interesting because if you look at the landscape right now, the tools we already have to break these protein bonds rely on drugs like venetoclax or certain hepatitis C inhibitors.
Right, those are the current standards.
Which brings me to a big point of pushback when I first started reading about this. If we already have FDA-approved drugs that can act as these chemical toothpicks, why bother with caffeine?
It's a great question.
Like, why go through the monumental effort of building this system from scratch just so I can use my espresso as a medical device?
Well, this raises an important point about the clinical realities of putting synthetic biology into a living human being. Let's look at venetoclax.
Okay.
When you use that as your trigger, you have to acknowledge what venetoclax fundamentally is. It is a highly potent anti-cancer medication. Its mechanism of action involves targeting a very specific family of proteins in the body called the BCL2 family.
And those BCL2 proteins aren't just in cancer cells, right?
Okay.
They are endogenous. I mean, they naturally occur everywhere in our bodies.
Yes. They are critical for normal cellular survival. In healthy cells, the BCL2 proteins basically act as a guard against premature cell death or apoptosis.
Oh, I see where this is going.
Right. So if you engineer a synthetic biology switch that requires venetoclax to operate, every single time you give the patient that drug to flip your synthetic switch, you are simultaneously attacking the natural survival mechanisms of all their healthy cells.
That is a severe friendly fire problem.
Exactly. You absolutely do not want your cellular pause button to double as a potent chemotherapy drug.
Yeah, that sounds like a therapeutic nightmare. You're trying to fine-tune an implant and you end up giving the patient massive off-target toxicity.
It defeats the purpose of precision medicine.
But what about the hepatitis C drugs, like the NS3A inhibitors? Because they are cancer drugs, right?
They avoid the friendly fire issue, sure, but they introduce a severe physical limitation. The molecules that make up those hepatitis C inhibitors are structurally very large and bulky.
Yep.
Because of their size and their chemical properties, they are notoriously difficult to wash out of a cellular system. Once they enter a cell and bind to their target, they tend to, well, overstay their welcome.
Which means your finely tuned switch essentially gets jammed.
Exactly. If the goal is a fully reversible system, something you can turn off at breakfast and have turn naturally back on by dinner time, you require a trigger molecule that diffuses into the tissue rapidly, does its job, and then clears out of the body at a predictable, swift rate.
And enter caffeine.
Enter caffeine. It is essentially the biological Goldilocks molecule for this specific engineering challenge.
Because it's everywhere.
It's incredibly cheap to synthesize, universally available, and from a medical perspective, we possess decades of deep pharmacokinetic data on it.
Right. We know exactly what it does in the human body.
We know the exact pathways it takes through the human liver, how efficiently it crosses the blood-brain barrier, its exact half-life in the bloodstream, and how it washes out of human tissue.
That makes so much sense.
And crucially, at normal physiological concentrations, like the kind you get from drinking coffee or tea, it is remarkably safe.
Here's where it gets really interesting to me, though. The why makes perfect sense. We want safety, fast clearance, cheap availability. But the how is what bends the mind.
Oh, it's fascinating.
Because millions of years of human evolution did not design our proteins to act as a mechanical light switch for coffee.
No, we definitely didn't evolve for that.
The scientists couldn't just find this in nature. They had to build a molecular machine that responds exclusively to caffeine. And to do that, they turned to artificial intelligence.
Yes. The convergence of artificial intelligence and physical biochemistry is arguably the most consequential technological leap we are witnessing this decade.
It's huge.
To understand how they constructed this, we have to look at the baseline ingredients they were working with.
So what was the foundation? What did they actually start with?
They began with a very specific pre-existing protein module known as Cosmo. To be technically precise, Cosmo is a specialized nanobody.
Let's quickly define nanobody for the listener, because that isn't the term you hear every day. How is it different from like a regular antibody that fights off a cold?
Standard antibodies, the kind our human immune systems produce, are quite large, complex, Y-shaped proteins. They are very effective, but they can be fragile and bulky to engineer. Nanobodies, on the other hand, were actually originally discovered in camelids animals like alpacas and llamas.
Wait, alpacas?
Yeah, their immune systems produce these incredibly tiny, stripped-down, highly stable antibody fragments. Scientists love them because they're tough, easy to manufacture, and they can fit into microscopic crevices that normal antibodies just can't reach.
So Cosmo is one of these tough, tiny alpaca-style proteins, but it's been specifically identified because it has a knack for grabbing onto caffeine.
Precisely. It's essentially a microscopic caffeine catcher.
Okay, got it.
And it possesses a very rare and highly useful structural quirk. When a molecule of caffeine floats by and slots perfectly into the binding pocket of a cosmoprotein, the entire protein physically shifts its shape.
Like a transformer.
Kind of.
Yep.
And that shape-shifting suddenly makes that specific cosmoprotein incredibly sticky toward other cosmoproteins. It initiates a process called homodimerization.
Okay, let's break that down. Homo meaning same, dimer meaning two parts. So two identical cosmoproteins snap together, but only when a caffeine molecule is sandwiched in the middle, acting as the structural keystone.
You got it. That mechanism was the raw material. But consider the engineering problem. The researchers did not want a system that sticks together in the presence of caffeine.
Right. They needed a chemically disrupted proximity system.
They needed it to fall apart.
Right. The molecular Velcro that tears apart on command. So how do you cross that bridge? How do you take a system whose natural instinct is caffeine makes us stick together and re-engineer it so caffeine makes us violently separate?
That is where the AI pipeline completely rewrites the rules of what's possible in a laboratory. The team utilized an AI framework known as Bindcraft. But to use Bindcraft, they first had to feed it data from another AI, AlphaFold.
I feel like we need to pause and really respect what AlphaFold is because it's hard to overstate its impact on biology.
It solved a grand challenge that had stumped the brightest minds for half a century, the protein folding problem. A protein starts out as a simple, one-dimensional string of amino acids dictated by DNA. But to function, that string has to fold up into a very specific, highly complex, three-dimensional origami shape.
And for decades, scientists knew the string, but they had absolutely no idea how it would fold.
Right. There's something called Levinthal's paradox, which notes that a typical protein could theoretically fold into so many different shapes that it would take longer than the age of the universe for a computer to randomly guess the right one.
Which is mind-blowing.
Yet inside your cells, proteins fold into their correct shape in milliseconds. AlphaFold essentially cracked the code. It uses deep learning to look at the one-dimensional string and predict, with atomic-level accuracy, the exact 3D shape that protein will take in the real world.
It's basically like having the master architectural blueprint for every biological machine on Earth.
Precisely. So the researchers took the AlphaFold blueprint of the cosmoprotein, and they fed that ultra-precise 3D map into the Bindcraft AI. And they gave the AI an incredibly complex prompt. They effectively asked it to invent a brand new, entirely artificial mini-protein, which they called a binder.
A completely new protein.
Right. They wanted this binder to stick powerfully to the outside of the cosmoprotein. But the crucial instruction was, make sure this artificial binder sticks to the exact same physical location where a second cosmoprotein naturally wants to bind when caffeine is present.
Oh, it's a microscopic game of molecular musical chairs.
I love that analogy. Walk us through how you're visualizing that.
Okay, so picture the cosmoprotein as a chair. In a cell environment where there is no caffeine around, the AI-designed binder protein is sitting comfortably in that chair.
Yes.
It's bonded right to the side of Cosmo. They are locked together in a stable state, but then the caffeine music starts. You drink your coffee, the caffeine molecule enters the cell, and it slots into Cosmo.
And remember, caffeine makes Cosmo desperately want to grab its twin, a second cosmoprotein.
But the AI binder is currently occupying the exact spot where the twin needs to attach.
Exactly. So what happens is a chemical eviction.
Wow.
The caffeine triggers such an intense thermodynamic attraction between the two cosmoproteins that they violently snap together. In doing so, they literally kick the AI binder out of the chair.
It's what chemists call a steric clash, right? right?
Yeah.
Two things just cannot occupy the same physical space.
That's right. So Cosmo grabs its twin, and the AI binder is forcefully ejected. The initial bond is broken. The proximity is disrupted.
It's an incredibly elegant physical solution. By using caffeine to trigger the dimerization of two cosmoproteins, they purposefully generate a competitive steric clash that crowds out the artificial binder.
And what is truly staggering is the timeline here.
Oh, yeah?
Before generative AI, a lab might have spent 5 years manually mutating proteins, testing millions of combinations blindly, hoping to find one that happened to fit that chair perfectly. The Bindcraft AI generated the exact sequence of amino acids to build that bespoke binder in a matter of computational hours.
It's generative AI, but
Oh.
not for text or images. It's generating physical reality. It invented a biological object that has literally never existed in the history of the planet, purely to play this microscopic game of musical chairs.
It shifts biology from a science of discovery to a science of pure design.
Which is thrilling.
However, as powerful as these in silico or computer-based models are, they are still approximations of the incredibly complex, noisy physics of the real world. The AI's first draft is rarely flawless.
So it wasn't a hole-in-one.
No, Bindcraft didn't just spit out one guaranteed winner. It provided the research team with 13 candidate binders. 13 distinct mini-proteins that the neural network calculated had a high probability of working.
Okay, so you have 13 microscopic candidates. You can't just line them up on a desk and look at them with a magnifying glass. How do you figure out which of the AI's guesses is the actual winner?
This is where the researchers had to transition out of computer science and back into the physical wet lab.
The messy stuff.
Exactly. They required a system to visibly confirm which of these 13 binders actually formed a strong bond with Cosmo in a living, breathing cell, and more importantly, which ones successfully fell off when caffeine was introduced.
And the source material details this really clever visual screening process.
Mm-hmm.
Instead of doing this in a sterile test tube, they performed the test inside living HeLa cells.
Yes. HeLa cells are fundamentally the workhorse of modern cellular biology.
Right.
They are an immortalized human cell line that has been cultivated in laboratories worldwide for over 70 years. Because they're so robust and well understood, they provide the perfect baseline environment for this kind of advanced microscopic imaging assay.
So I'm looking through the microscope. What am I actually seeing? Like, how do they make these invisible proteins visible to the human eye?
They utilized fluorescence microscopy, which relies on proteins that literally emit light.
Cool.
They took the cosmoprotein and, at a genetic level, fused it to a protein called EGFP. EGFP stands for Enhanced Green Fluorescent Protein.
Jellyfish, right?
Yes. It's a molecule originally isolated from jellyfish that glows a brilliant, vibrant green when you shine a specific wavelength of light on it. But they didn't just let this green cosmoprotein float aimlessly in the cellular fluid. They added a tiny molecular anchor to its genetic code.
Oh, so that anchor physically ties the green cosmoprotein to the inner surface of the cell's outer skin, the membrane.
Exactly. So if you look at that cell under a microscope, the outer border, the entire circumference of the cell, is traced in a sharp, glowing green line.
Got it.
Then they took the 13 AI-designed candidate binders and they genetically fused them to a completely different fluorescent protein called M-cherry.
Which, I'm assuming from the name, glows red.
A very bright red. Now, visualize the physics of the cell interior. If an AI binder does not work if it fails to stick to the green cosmoprotein on the membrane, it just diffuses randomly throughout the liquid interior of the cell, which is called the cytosol.
So you'd see a green ring around the outside, and the entire inside of the cell would just be a hazy, uniform red fog.
But if the AI's design was successful, if the red binder strongly grabs onto the green cosmoprotein,
then all that red protein gets pulled out of the liquid interior. It gets dragged directly to the cell membrane where Cosmo is anchored.
right?
And because green light and red light are overlapping right on top of each other, the entire boundary of the cell suddenly glows a brilliant yellow-orange.
It is an incredibly clean binary visual readout. You just look through the lens for the yellow ring. So they methodically tested all 13 AI candidates inside these living cells.
So what was the verdict? Did the AI nail it on the first try?
Most of the candidates failed, actually.
Really?
Yeah, the complex fluid dynamics of a real cell are really hard for a computer to perfectly predict. But one of the candidates, binder number 3, which they just called B3, was a spectacular success.
Number 3.
When they expressed the B3 protein in the HeLa cells, the hazy red interior vanished, and the cell membrane lit up with that beautiful yellow-orange band. The B3 binder was physically being pulled out of the fluid and locked onto the cosmoprotein.
The molecular Velcro was attached. The AI binder was sitting firmly in the musical chair.
Exactly.
But that's only half the battle, right?
Yeah.
Now for the ultimate test, the coffee drop.
The researchers pipetted a minuscule concentration of caffeine into the Petri dish housing the cells. And almost instantaneously under the microscope, that bright yellow-orange ring around the cell membrane dissolved.
Wow.
The green anchor remained, but the red glow rushed back out, dispersing evenly into the hazy interior of the cell.
The caffeine stopped the music.
Yes.
The cosmoproteins snapped together, grabbing their twins, and B3 got violently kicked out of the chair, tumbling back into the cytoplasm. It worked. They successfully built a microscopic, caffeine-operated switch.
They absolutely did. However, the initial prototype wasn't flawless.
Oh, the inevitable catch. What was wrong with the B3 binder?
In any kind of engineering, biological or mechanical, you have to contend with the signal-to-noise ratio. The B3 binder definitively detached when caffeine was added. That's the desired signal. But the problem was the noise occurring before the caffeine was ever introduced. The baseline bond between the AI's B3 protein and the cosmoprotein wasn't quite strong enough to hold steady in the chaotic environment of the cell.
Because of that molecular mosh pit we talked about earlier,
Yeah.
everything is constantly vibrating and crashing.
Exactly. Because the bond was slightly weak, a significant portion of the red B3 protein was constantly getting knocked off the membrane and swimming around in the cytoplasm, falling off and reattaching dynamically. In engineering terms, they had built a leaky system.
A leaky switch.
Yeah.
It's like a garden hose that drips even when you've cranked the handle all the way to the off position.
That's a great way to put it.
And if your goal is to eventually use this inside a human body to control powerful medicines, you cannot afford a drip. You need it completely securely, 100% locked until the patient takes that sip of coffee. So how do they fix the leak? They needed a much stronger bond.
To eliminate the leak, they had to upgrade the binder. They had to introduce mutations to its genetic code to alter its shape and make it grip the cosmoprotein tighter.
Right.
But again, going back to the lab and manually testing thousands of random mutations would take months, if not years, of pipetting. So they returned to the supercomputer. They moved the project back in silico.
And just to clarify the terminology for everyone, in silico means the experiment is performed on a computer via simulation, as opposed to in vivo, which is inside a living animal, or in vitro, which is in a glass test tube.
Correct. They employed a massively powerful computational biology suite called Rosetta. Rosetta is specifically designed to predict how making tiny single amino acid changes to a protein structure will alter its overall physical stability and energy.
Okay.
They instructed the software to systematically simulate changing every single amino acid on the surface of the B3 protein, just tweaking the exact spots where it physically touches the cosmoprotein.
And what exactly is the computer looking for in these simulations? Like, how does an algorithm actually know if a molecular bond is physically tighter?
The software calculates a very specific thermodynamic metric called the Gibbs free energy change. In biochemistry, it's often represented by the symbol delta delta G.
Okay, let's slow down and unpack Gibbs free energy, because that sounds like a college physics exam waiting to happen.
It is the bedrock of physical chemistry. To understand it, you just have to remember that in thermodynamics, physical systems naturally prefer to exist in their lowest possible energy state.
Okay.
Think of a heavy boulder resting at the top of a steep hill. It possesses a lot of potential energy, and it's somewhat unstable. If you nudge it, it naturally rolls down into the valley, coming to rest at the bottom where its potential energy is lowest. It's much more stable down there.
Right. It's not going to spontaneously roll back up the hill without a huge input of outside energy.
Exactly. Molecules behave the exact same way. When two proteins bind together, they want their physical shapes and electrical charges to align so perfectly that they fall into a deep, stable thermodynamic valley. A negative delta G value in these calculations indicates that a specific mutation allows the two proteins to settle into an even lower, more comfortable, more stable energy state together.
So they fit together more snugly. The molecular Velcro gets denser and tighter.
Yes, exactly. But the sheer computational horsepower required to calculate this is staggering.
I bet.
The researchers weren't just looking at static shapes, they were running molecular dynamic simulations on high-end GPUs at a computational speed of 202 nanoseconds per simulated day.
Wait, 202 nanoseconds per day? That sounds infinitesimally slow.
Well, in the realm of all atom simulations, it is actually blindingly fast. You have to realize what the computer is doing. It isn't just drawing a picture.
What's it doing?
It is mathematically tracking the movement, the electrical repulsion, the van der Waals forces of every single individual atom in that protein complex. It is simulating the microscopic wiggling, jiggling, and vibrating of the molecules at femtosecond intervals, quadrillionths of a second.
Wow. It is wild to think about. Decades ago, finding a tighter binding protein would have been a lifetime of tedious manual labor for a team of PhD students, just mutating genes blindly and praying one stuck better.
Oh, easily.
Now, it's a few days of letting a graphics card grind through the laws of physics.
Yeah.
So after all that math, what was the result? Did Rosetta find the magic mutation to fix the leak?
It did. Out of all the thousands of simulated permutations, one specific mutation stood out with an incredibly favorable thermodynamic profile.
Nice.
It was a single substitution located at position 44 of the protein chain. The software suggested swapping out an amino acid called arginine for a different one called valine.
Okay. Arginine for valine.
Right. They designated this upgraded version the R44V mutation. The physics engine predicted this tiny swap would profoundly stabilize the grip between the two proteins.
But did it translate from the computer screen back to the real world? Like, did it work in the glowing HeLa cells?
It worked beautifully. When they expressed the R44V variant in the living cells, the fluorescent leak was almost entirely eradicated.
That's amazing.
To put numbers on it, they measure the ratio of the red protein firmly stuck to the membrane versus the amount lazily floating in the liquid. With the original B3 binder, that ratio was 1.4.
Okay.
With the new R44V mutation, it jumped to 1.8. It was a dramatically tighter, more secure lock.
The leaky faucet was completely shut off.
Yes. But the brilliant part is that even though the binder was gripping the cosmoprotein much tighter, it did not lose its sensitivity to the caffeine trigger. When they introduced caffeine to the upgraded system, the disruption was still incredibly fast.
Let's talk about that speed test, actually, because the data on this is wild. How fast are we talking?
The kinetic data is truly impressive for a biological system. From the exact moment the caffeine molecule diffuses into the system, it takes an average of merely 2.0 minutes for the artificial protein to completely detach from Cosmo. And what is equally vital for medical applications is the reversibility. If you wash the caffeine away, which perfectly mimics the human liver filtering caffeine out of the bloodstream over time, it takes only 2.4 minutes for the upgraded B3 protein to locate the Cosmo protein and reattach.
2 minutes. That isn't sluggish biological time. That is near real-time mechanical control.
It really is.
You drink a cup of coffee, it gets absorbed in your stomach, and within minutes of it hitting your bloodstream, your cellular machinery shifts gears.
It represents the exact kind of rapid, highly tunable, reversible control that synthetic biologists have been trying to achieve for decades, which naturally brings us to the most critical part of this entire discussion.
Right.
The ultimate reason why this research was conducted.
The so what factor. We have spent all this time geeking out over molecular musical chairs, fluorescent jellyfish proteins, and supercomputer simulations. We have successfully built a microscopic switch that flips on and off with staggering precision when exposed to caffeine. But what can we actually do with it?
Well, a switch is ultimately only as valuable as the machine it is wired to. And in the latter half of this research, the team took their CODS system and wired it into 3 incredibly diverse, highly impactful biological applications.
Okay, let's start with application 1, which involves controlling the actual DNA code of the cell. They call it transcriptional reprogramming, which essentially means turning specific genes on or off at will.
This serves as a fundamental proof of concept for the whole system. The ability to dictate exactly when a gene is active or silent is the holy grail of genetic engineering.
Why is that so hard usually?
Normally, if a scientist inserts a therapeutic gene into a human cell, it's generally just on. It constantly reads the code and turns out whatever protein it's designed to make.
It's like having a high-powered light fixture wired directly into the city's power grid, but no one bothered to install a light switch on the wall. It's just blazing 24/7.
Exactly. To build that missing wall switch, the researchers took their caffeine system, the Cosmo protein, and the upgraded B3 binder, and physically fused them to a very well-established gene expression tool known as the TTA system, which relies on a protein called TET-R.
Okay, let's break down the mechanics of that for everyone.
Yeah.
How does a molecular switch physically stop a strand of DNA from being read?
The easiest way to visualize a gene is to think of it as a recipe written in a massive cookbook.
Okay.
In order for the cell to read that recipe and bake the cake, which in this case is producing a protein, a complex molecular machine called RNA polymerase has to land on the page and physically scan across the letters.
That makes sense.
The TET-R system they use is basically a two-part reading machine. One half acts as a molecular anchor. Its sole job is to find the start of the recipe and lock onto the DNA.
And the other half?
The other half is the engine that actually drives the reading process and recruits the polymerase.
So logically, if the engine isn't physically attached to the anchor, the recipe never gets read. The machine is broken.
Precisely. And this is where the researchers' engineering shines. They took the DNA anchor and they genetically fused the cosmoprotein to it. Then they took the engine part and they fused the AI-designed B3 binder to it.
Oh, I see the architecture.
Right.
As long as there is no caffeine floating around in the cell, the B3 binder naturally sticks to the cosmoprotein. Because they stick together, the engine is firmly attached to the anchor, the molecular reading machine is fully assembled, sitting on the DNA, and the gene is actively being read.
Correct. In their experimental setup, they hooked this machine up to a gene that codes for a protein called SEEP.
Let's define SEEP really quickly.
SEEP stands for secreted alkaline phosphatase. It's essentially a reporter protein. The cell produces it and then actively pumps it out into the surrounding fluid.
So it's easy to track.
Exactly. Scientists love using it because you don't have to break the cell open to see if the gene is working. You just sample the fluid around the cell and measure how much SEEP is floating in it. They also ran the test using luciferase, which is the exact same enzyme that allows fireflies to glow in the dark. Without caffeine, the cells happily churned out SEEP and luciferase. The gene was locked in the on position.
But then, you introduce a tiny amount of caffeine to the environment. The molecular musical chairs process initiates. The caffeine slots into the cosmoproteins violently dimerize, and the B3 binder gets forcefully evicted from its seat.
And because the B3 binder is attached to the engine violently separates from the DNA anchor.
Wow.
The reading machine is physically snapped in half. The gene expression immediately halts.
And the numbers on this are incredibly stark. How much caffeine did it take to actually shut this genetic assembly line down?
An astoundingly small amount. With a concentration of just 100 nanomolar of caffeine, the expression of the reporter proteins plummeted by 20.5-fold.
A 20-fold drop from just a microscopic whisper of caffeine. It proves the system is an incredibly tight, reliable off-off switch. And because we know the thermodynamics from the supercomputer, when the caffeine eventually washes away, the engine finds the anchor, reattaches, and the gene turns right back on. You have achieved totally reversible, on-demand fluid control over a cell's operating system.
It is a remarkable technical achievement. But as you noted earlier, turning off a glowing jellyfish protein or measuring SEEP in a dish is ultimately a parlor trick to prove the plumbing functions correctly.
Right. Tumors don't care if a cell glows.
Exactly. To actually impact a disease, you need the switch to trigger something aggressive, something lethal, which transitions us perfectly into application 2. The researchers wired the coffee switch to a mechanism called pyroptosis.
Pyroptosis, just the etymology of that word is intense. Pyro meaning fire, ptosis meaning falling. Fiery falling.
The aggressiveness of the name is entirely warranted by the biology. Pyroptosis is a highly specialized, intensely inflammatory form of programmed cell death.
How is that different from normal cell death? Because our cells die all the time, right? Like skin cells flake off, old red blood cells get recycled. We aren't spontaneously combusting all day.
When a normal cell reaches the end of its natural lifespan or sustains irreparable DNA damage, it undergoes a very quiet, orderly process called apoptosis.
Apoptosis, okay.
During apoptosis, the cell essentially shrinks down, cleanly packages its internal contents into tiny, neat vesicles, and allows local cleanup cells to quietly consume it without disturbing any of the neighboring tissue. It's a highly polite way to die.
So it's peaceful.
Pyroptosis is the exact opposite. It is a cell pulling the pin on a grenade. The cell rapidly swells up, violently bursts its membrane open, and spews massive amounts of highly inflammatory chemicals into the surrounding tissue to scream at the immune system. Look over here, we are under attack. Something is terribly wrong.
Wait, why on earth would doctors want to trigger a biological grenade explosion on purpose?
Yeah.
It sounds inherently dangerous.
Because in the context of treating solid tumors, getting the immune system's attention is the hardest part of the battle. Many solid tumors are what oncologists call cold.
Cold tumors.
Yes. The cancer cells actively manipulate their local microenvironment to suppress immune activity. They create a quiet, invisible bubble where T cells simply don't attack.
They're hiding.
If a doctor could engineer a small population of cells to infiltrate that tumor and then undergo pyroptosis, they could shatter that quiet bubble. The fiery explosion of inflammatory signals would instantly convert the cold tumor into a hot one, drawing a massive swarm of the body's natural immune defenders right to the site of the cancer.
Okay, that makes incredible strategic sense. It's a targeted biological beacon flare. So how did the researchers wire their caffeine switch to act as the detonator for this explosion?
To build the detonator, they hijacked this specific protein that naturally executes pyroptosis in the human body. It is a protein called gastromin D.
Gastromin D. The mechanical structure of this protein is fascinating, isn't it?
Yeah, it truly is. Gastromin D is essentially constructed of two distinct halves connected by a flexible molecular hinge. One half of the protein is the pore-forming domain. This is the actual explosive payload. If this pore-forming half ever gets loose, it aggressively drives itself like a spike into the cell's outer membrane, polymerizes with other gastromin spikes, and punches massive gaping holes in the cell wall, causing the cell to explode.
So if we all have gastromin D inside our cells right now, what stops us from exploding at random?
The other half of the protein, the second half, is known as the autoinhibitory domain. Its entire structural purpose is to physically wrap around the explosive part, tightly hugging it to mask its lethal properties and prevent it from doing any damage.
Like a safety pin.
Right. In a natural biological scenario, an invading bacteria has to trigger a specific enzyme called a caspase, which comes along like a pair of molecular scissors, snips the hinge, and unleashes the explosive part.
Oh, I see the play here.
Yeah.
The researchers removed the natural biological hinge and they replaced it with the artificial CODS system.
Exactly. They engineered a synthetic, customized version of gastromin D. They took the explosive, pore-forming payload and tethered it directly to the AI-designed B3 binder. Then they took the protective, autoinhibitory hugger and tethered it to the cosmoprotein.
So as long as there is no caffeine in the system, the B3 binder holds tight to the cosmoprotein.
Yeah.
Because they are locked together, the explosive part of gastromin D is being tightly held and neutralized by the protective part. The cell is perfectly safe. The safety pin is firmly locked in the grenade.
Correct. But the very moment a molecule of caffeine diffuses into that cell,
The mechanical switch flips. The
Yeah.
caffeine hits the cosmoprotein, forcing it to dimerize with its twin. The AI binder gets violently kicked away via steric clash. The protective half of the protein lets go. The explosive payload is suddenly naked, unleashed, and active.
and it immediately zeroes in on the cell membrane to start punching holes. The physical evidence presented by the researchers to prove this is absolutely stunning. They provided time-lapse microscopy of this precise event happening in real time.
I saw those images in the source material. It is pure microscopic drama. Within just 60 minutes of dropping the caffeine onto the cells, you can visibly see the cell membranes start to warp.
Yeah.
They form these massive protrusions they call pyroptotic bubbles. The cells literally look like they're boiling from the inside out.
Those boiling bubbles are the exact locations where the unleashed gastromin D is destroying the structural integrity of the membrane. The internal pressure of the cell is pushing the weakened, leaking membrane outward.
And within 3 hours, it's game over. The cells completely rupture. They literally pop, leaking all their internal contents out into the surrounding fluid. To prove they popped, the researchers measured the release of an enzyme called LDH.
Which is a crucial detail. LDH, or lactate dehydrogenase, is an enzyme that helps cells metabolize sugar. Under normal conditions, LDH is strictly locked securely inside the cell. It's too big to just leak out.
So if it's outside,
Right. If a scientist finds high levels of LDH freely floating in the fluid outside the cell, it is the ultimate biochemical proof that the cell's outer membrane has been catastrophically blown apart.
it's forensic evidence of a cellular explosion. And this entire violent cascade was activated by nothing more than a localized drop of caffeine.
Imagine the clinical implications of this down the line. You could inject a patient with engineered therapeutic cells that are designed to silently navigate the bloodstream and embed themselves deep inside a hard-to-reach solid tumor.
Yeah.
They sit there quietly. Then, simply by having the patient drink a predetermined amount of coffee, you remotely detonate those cells.
Wow.
They burst, releasing a highly localized payload of chemotherapy drugs right into the tumor core, or as we discussed, triggering a massive immune system flare exactly where you want it, with zero collateral damage to the rest of the body.
That is wild. It's a remote-controlled smart bomb operating inside the human body.
Mm-hmm.
But as visually dramatic as the exploding cells are, I want to transition to the third and final application.
Yes.
Because when I was reading through the source material, Application 3 struck me as the absolute holy grail of this paper. This isn't just a future hypothetical. This is a technology that could fundamentally change how oncologists operate in hospitals tomorrow. We are talking about taming the cure or the precision tuning of CAR T-cell therapy.
I agree completely. In the landscape of modern medicine, CAR T-cell therapy is arguably the most miraculous, paradigm-shifting advancement in oncology of the last 50 years. But it possesses a terrifyingly dark side that desperately requires an elegant engineering solution precisely like this.
Let's lay the historical and biological groundwork for the listener. What exactly is a T-cell, and what makes a CAR T-cell so special?
In the simplest terms, T-cells are the elite special forces soldiers of your innate immune system. They constantly patrol your bloodstream and your tissues, looking for cells that have been infected by a virus or cells that have mutated and become cancerous.
Okay.
When a normal T-cell finds a threat, it locks onto it, secretes toxic chemicals, and obliterates the diseased cell.
But if our T cells naturally kill cancer, why do people get cancer in the first place?
Because cancer is incredibly sneaky. It evolves. Tumors develop complex mechanisms to essentially become invisible to normal T-cells.
They hide.
Exactly. They hide the molecular flags that say, I'm sick, or they produce chemicals that force the T-cells to fall asleep when they get too close. This is where CAR T-cell therapy changed the world.
How does it work?
In this therapy, a doctor extracts a sample of the patient's own naturally occurring T-cells. They take those cells into a highly specialized laboratory, and they use a harmless virus to edit the T-cell's DNA.
So they are genetically upgrading the soldier.
Exactly. They engineer the T-cell to produce a completely artificial synthetic protein on its surface called a chimeric antigen receptor, or a CAR-R.
An chimeric antigen receptor. Chimeric, referring to the mythic Greek beast made of different animal parts, like a lion's head, a goat's body. In this case, it's a synthetic receptor pieced together from different biological domains. It's basically a custom-built radar dish that you weld onto the surface of the T-cell.
It is a perfect description. This synthetic radar dish is explicitly molecularly tuned to recognize one specific protein that is known to exist on the surface of the patient's specific cancer cells.
So it can't hide anymore.
Right. Because the radar is synthetic, the cancer's normal tricks to hide from the immune system no longer work. When doctors infuse these engineered CAR T cells back into the patient's bloodstream, they act as guided munitions.
Wow.
They hunt down the cancer with terrifying, relentless efficiency. This therapy has legitimately cured patients with advanced terminal B-cell leukemias and lymphomas who had failed every other treatment and had weeks left to live.
It sounds like an absolute miracle cure. So where does the dark side come in? Why do we need an emergency switch for a miracle?
The profound danger lies in the fact that these engineered immune cells can sometimes work too efficiently.
Too efficiently?
Yeah. When a CAR T-cell locates a cancer cell and begins its attack, part of its programming is to rapidly multiply, creating thousands of copies of itself, and to release massive amounts of inflammatory signaling proteins called cytokines.
And cytokines act as a chemical alarm, calling every other immune cell in the body to join the fight.
Exactly.
Which, on paper, sounds like exactly what you want to happen. You want to overwhelm the cancer.
You do, up to a point. But in roughly half of all patients, this intense activation triggers a runaway, cascading biological chain reaction called cytokine release syndrome, or CRS. The immune system goes into a terrifying, uncontrolled overdrive.
Oh, no.
It stops just attacking the cancer and starts attacking everything. The patient experiences a skyrocketing, dangerous fever. Their blood pressure plummets to life-threatening levels, fluid fills their lungs, their organs can begin to systematically shut down.
That's horrifying.
It gets worse. Furthermore, it can cross the blood-brain barrier and cause severe neurotoxicity, leading to brain swelling, delirium, and fatal seizures.
So the cure literally becomes as deadly as the disease. The engineered T cells are successfully annihilating the cancer, but the inflammatory crossfire is killing the patient.
Precisely. And the agonizing dilemma for an oncologist is that if a patient crashes into severe cytokine release syndrome, the medical toolkit is frighteningly bare.
What can they even do?
They can administer massive doses of broad-spectrum steroids or specialized immunosuppressants to try and calm the storm, but doing so often completely kills off the engineered CAR T cells.
So you lose the cure.
Exactly. You save the patient from the storm, but you permanently end their cancer treatment, allowing the tumor to roar back. What the field of oncology desperately, urgently needs is an emergency brake, a temporary reversible pause button that forces the CAR T cells to stop attacking for just a few hours to let the patient's body cool down.
Enter the caffeine-operated dissociation system.
The researchers took their CRDS architecture and utilized it to construct what is known as a split CAR.
A split CAR. Okay, so instead of one big, solid synthetic radar dish protruding from the surface of the T cell, they essentially cut the radar dish in half.
Right. To understand the split, remember how the standard CR is built. The external part that sticks out of the cell physically grabs onto the cancer cell, and the internal part, which dips inside the T cell, acts as the ignition wire.
Boy.
When the outside grabs the cancer, it yanks the wire, sending the attack signal to the T cell's nucleus. In the split FIR design, the engineers physically severed the external grabber from the internal ignition wire.
Oh, and let me guess exactly how they bridged that gap. They attached the cosmoprotein to one of the severed halves, and they attached the AI's B3 binder to the other half.
That is exactly the architecture. So long as there is zero caffeine circulating in the patient's bloodstream, the cosmoprotein and the B3 binder naturally snap together with a tight grip.
Making it whole again.
Because they're joined, the two severed halves of the split FIR are functionally fused back into a single operational unit. The radar dish is fully assembled.
So it works like normal.
Right. The T cell perfectly targets the cancer. In the paper, they specifically tested this against a cancer target called the CD19 protein on Raji lymphoma cells, and it destroys the tumor just like a normal SAR T cell would.
But if the patient is lying in the ICU and the monitors start blaring, if their temperature spikes, their blood pressure crashes, and the clinical team realizes a lethal cytokine release syndrome cascade is beginning.
The doctors do not have to resort to toxic, therapy-ending immunosuppressants. They simply administer a highly specific, calculated dose of caffeine.
The switch flips. The caffeine rushes into the bloodstream and diffuses into the T cells. It binds to the cosmoproteins. The cosmoproteins immediately dimerize, violently kicking the AI binder away.
And then
The external grabber on the outside of the cell is instantaneously physically disconnected from the ignition wire on the inside. The radar dish is broken, the T cell loses its signal, stops attacking, and goes to sleep. The emergency brake has been successfully pulled.
And the quantitative data they generated to prove this is phenomenal. They spent a lot of time testing different structural permutations of exactly where to cut and join the CRR, but their most optimal configuration, which they designated the 1B plus 2B setup, was remarkably suppressive.
Let's get into the hard numbers of the data because it really drives home how effective this is.
To accurately measure exactly how angry and active the T cells were, the researchers tracked the cellular production of a specific signaling molecule called IL-2, or interleukin-2.
Look.
When a T cell is in full aggressive attack mode, it pumps out massive quantities of IL-2. In their optimized 1B plus 2B split CR configuration, when there was no caffeine present, the T cells were fiercely attacking the lymphoma cells, generating extremely high levels of IL-2.
But the moment they introduced the caffeine to the environment,
The IL-2 production crashed by a staggering 26.9-fold.
26.9-fold, that isn't just a minor dimming of the lights, that is slamming the emergency brake to the floor and coming to a violent screeching halt. The T cells fundamentally ceased their attack.
It serves as an incredibly robust, deeply reliable off switch. And crucially, because the mechanism relies on chemical dissociation, physically breaking a bond, the effect is virtually instantaneous.
Right, because it's the physical separation.
The exact moment the caffeine molecule reaches the receptor, the complex splits. The clinical team doesn't have to sit there nervously for 4 hours waiting for a biological pathway to slowly clear out a drug. The physical connection transmitting the attack signal is just gone.
Now, as I was reading through this specific section of the data, looking at the charts, I actually had a massive moment of pushback.
Oh, really?
Yeah. A real, wait a minute, this makes no sense moment regarding what I'll call the dosage debate.
Okay, let's explore that. What caught your eye?
So I looked closely at the concentration numbers. The data shows that the IC50 of the system, which in pharmacology means the exact concentration of caffeine required to successfully turn off 50% of the active CRT cells, is around 34.3 nanomolar.
That is correct. 34.3 nanomolar is an exquisitely tiny, microscopic concentration of caffeine.
Exactly. It is vanishingly tiny. But then I cross-referenced that with the pharmacokinetic data of normal human caffeine consumption. The researchers explicitly note that if an average adult drinks just a single standard cup of coffee, their blood caffeine level quickly peaks at around 10 micromolar.
Yes, roughly 10 micromolar is a standard post-coffee physiological baseline.
But hold on, 10 micromolar is literally almost 300 times higher than 34.3 nanomolar. The sheer amount of caffeine flooding your veins from one simple breakfast latte is astronomically higher than the microscopic amount needed to completely shut down the synthetic cancer treatment.
That's true.
So my immediate thought was, isn't this system fatally flawed? Won't a cancer patient accidentally turn off their life-saving $1 million immunotherapy just by eating a piece of coffee cake or having a morning espresso? Isn't the mechanism vastly too sensitive?
That is a brilliant catch, and from a traditional pharmacological perspective, it is a completely logical concern. You usually want the dose of your trigger to match the sensitivity of the receptor. But what is truly fascinating about this specific engineering challenge is that the researchers deeply considered this, and they view this extreme hypersensitivity not as a bug, but as the central vital feature of the entire system.
Wait, how can it possibly be a feature that a tiny sip of coffee breaks your $1 million medicine?
You have to view it through the lens of the specific clinical goal.
Yeah.
You are attempting to halt a raging, potentially lethal immune storm inside a crashing patient.
Okay. Yeah.
You want that emergency brake to be incredibly easy to pull, and vitally, you want it to stay pulled until the physiological danger has completely passed. Let's imagine a hypothetical scenario where the system was engineered to be less sensitive, say, if it actually required a full 10 micromolar of caffeine to flip the switch to O.
Okay, so if the threshold was much higher.
In that scenario, the patient would drink a coffee, hit 10 micromolar, and the T cells would pause. But the human liver is incredibly efficient at clearing caffeine.
Oh, I see.
Within an hour or two, their blood concentration would naturally dip to 9 micromolar, then 8 micromolar. It would drop below the operational threshold. The T cells would violently wake back up, and the cytokine storm would resume.
Wow, so you'd be in trouble quickly.
To keep the brake applied, the patient would literally have to be hooked up to a continuous IV drip of pure caffeine, or be constantly chugging espresso every single hour around the clock, which brings its own massive cardiovascular toxicities.
Oh,
Yeah.
I see the kinetic strategy now. Because the human body naturally filters and metabolizes caffeine over time, you're playing against a ticking clock.
Exactly. Caffeine possesses a biological half-life in the human body of roughly 4 to 6 hours. Because the CODS system is engineered to be so incredibly fundamentally sensitive, because its off threshold is a mere 34 nanomolar. A single, standard, perfectly safe oral dose of coffee pushes the initial blood concentration so profoundly high above the required threshold that it buys the clinical team a massive window of time.
So as the liver slowly, steadily clears the caffeine out of the blood over the next 12 to 14 hours, the systemic concentration drops from 10 micromolar down to 5, down to 1, down to 0.5, but it stays safely, securely, well above that tiny 34 nanomolar tripwire the entire time.
Precisely. You do not need a complex, continuously monitored IV drip of an experimental synthetic chemical. You literally just hand the feverish patient a standard cup of coffee.
That's wild.
And because the molecular switch is so incredibly sensitive to the molecule, that one single cup provides enough of an area under the curve to flip the switch and guarantee the T cells remain fast asleep for half a day while the body's inflammatory markers safely reset.
That is just brilliant biological engineering. The patient starts experiencing a life-threatening fever. The nurse hands them a coffee. The engineered T cells sleep for 12 hours while the patient's physiology recovers.
It's elegant.
And then, once the liver finally clears that very last lingering nanomolar of caffeine, the synthetic receptors automatically reassemble, the radar dish powers back up, and the cancer fight seamlessly resumes. No complex toxic pharmacology required.
It represents the absolute pinnacle of accessible, non-toxic, easily administered safety mechanisms in advanced genetic medicine.
So as we wrap up, let's take a step back and really look at the narrative arc of the science we've just traveled. We started with a foundational concept, the idea that we could move beyond just using chemical glue to stick biology together and instead harness the physics of molecular musical chairs to precisely pull proteins apart.
Right, the evolution from chemically induced proximity to chemically disrupted proximity.
And because manually finding a molecule to wedge two proteins apart was too thermodynamically difficult to do by simple trial and error, we outsourced the problem to artificial intelligence.
We did.
We fed a neural network the atomic blueprint of an alpaca-derived caffeine-catching protein. And the AI hallucinated into existence a bespoke, entirely artificial partner protein that is guaranteed to get violently kicked out of its seat the exact moment caffeine shows up.
Utilizing the Minecraft generative pipeline to invent Binder 3.
Then we visualized that invisible math. We watched inside living human cells as glowing red proteins were physically dragged to the green boundary of the cell membrane, and we watched them scatter back into the fluid like a flock of startled birds the second the coffee hit.
A beautiful experiment.
We leveraged the raw horsepower of modern GPUs to simulate the physical vibrations of individual atoms, discovering that one magic mutation, R44V, that tightened the molecular grip and stopped the switch from leaking.
Taking a process of laboratory evolution that used to span years and compressing it into mere days of thermodynamic computation.
And finally, we took that microscopic AI-designed switch and we hardwired it directly into the most critical machinery of human life. We used it to slice genetic reading machines in half, pausing the expression of DNA. We used it to pull the safety pin on a cellular grenade, detonating rogue cells on command to sound an immune alarm. And most profoundly, we used it to put a temporary leash on the most powerful cancer therapy ever invented, turning an ordinary cup of coffee into a life-saving emergency brake to rescue patients from lethal cytokine storms.
It truly is a masterclass in modern synthetic biology. It definitively demonstrates that we are no longer just passive observers discovering how biology works. We are actively, purposefully engineering it from the atomic level up with a degree of precision that was literally unimaginable a decade ago.
It really makes you pause and reevaluate everything you think you know about medicine.
And if you extrapolate this technology forward, it leads to a concept that completely breaks down our traditional barriers of what medicine even is.
How so?
Think about what we've just discussed. AI mapping a protein to respond flawlessly to caffeine, but caffeine is ultimately just one of thousands of distinct molecules present in our daily diet. If the supercomputers can design a bespoke switch for coffee, what is stopping them from doing this for anything else?
Oh, well, I see where you're going with this.
Could we eventually utilize generative AI to map out the unique chemical signatures of an entire specific diet? Consider the distinct, complex phytochemicals present in a bowl of raw spinach, or the highly specific flavonoids found in a square of 80% dark chocolate.
That's fascinating.
What if we engineer our body's internal medicine, our therapeutic immune cells, our implanted gene therapies, our metabolic regulators, to only activate or only deactivate based precisely on what we consume for lunch?
That is an incredibly profound thought. The idea that pharmacology and standard nutrition could eventually merge into the exact same scientific discipline.
Exactly.
Your future doctor's prescription isn't a plastic orange pill bottle. It's a very specific, chemically targeted grocery list designed to operate your engineered cellular hardware. The rigid boundary between our mundane daily diet and advanced medical intervention is suddenly completely blurring.
We are rapidly approaching an era where the everyday food we eat literally functions as the software code to program our augmented biology.
It really is a brave new world. So I want to thank you for joining us on this deep dive. We have covered an immense amount of ground today, from the physics of AI supercomputers to the violent beauty of exploding cells.
It's been a pleasure.
But the next time you sit down at your kitchen table, pour yourself a mug of coffee, and take that first warm sip, I highly encourage you to think twice about what is happening on a molecular level. Because you aren't just waking up your brain. You might just be holding the remote control to the future of human medicine. Usually when we talk about a medical diagnosis, there's this expectation of mechanical precision like reading a clean, binary X-ray. But maybe the future of medicine isn't a rigid X-ray at all. Maybe it's just a really good cup of coffee. Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.